Hello, my name is Abbey Wiggins, and today I'm going to be talking about how annual climatic variation affects plant biomass in tidal wetlands. I work under Dr. Julia Cherry. So before I get into it, I'm going to give a little overview of what I'm talking about today. I'll be talking about wetlands and why we care about them, some climate stressors on wetlands and meteorological data, the methodology of the study, and then some conclusions and implications. So tidal wetlands. They offer very important ecosystem services. They can cycle nutrients from pollution, such as nitrogen and phosphorus, which can help sustain other ecosystems. They offer storm surge protection, and with many of the hurricanes we've been seeing recently, they can help mitigate some of the damage from those storm surges. They can also permanently store carbon, which is important for climate change implications, and they are hubs of biodiversity. So, in this study, what I'm going to be focusing on today is plant biomass, and more specifically, above-ground plant biomass. So that refers to the standing vegetation of plants, which is gathered above the soil surface. And so with above-ground biomass, what I'm really getting at is an indirect measure of plant productivity. Plant productivity is a plant's ability to produce biomass, and it's commonly used as a measure of tidal wetland health and success. And so with above-ground biomass, we're really sorting those into two kinds of plants, which are C3 and C4 plants. And so this refers to a plant's photosynthetic pathway. And a C3 plant, when it's experiencing water stress or hot and dry conditions, it can close its stomata, which inhibits its ability to undergo photosynthesis. With a C4 plant, its photosynthetic pathway has an extra layer of insulation, so when it closes its stomata, it can still undergo photosynthesis in the bundle sheath because it can produce a 4-carbon concentrate. And so with climatic conditions, some of the climate change stressors that we're expecting to see in future years and that we're currently seeing are increased CO2 levels in the atmosphere, which contributes to global warming, which then can increase the sea level and contribute to sea level rise, which is a particularly tough stressor on tidal wetlands because they are coastal habitats. We also expect to see more frequent extreme storms and then more highly variable climatic conditions. So we expect ambient conditions to become more deviant from ambience. And so that's kind of what we're getting at with this study here, and that we want to see how something like annual climatic variation could affect the plant biomass in marshes and how that could impact their resilience to these other climate change stressors that we see. And so the study meteorological parameters that we're going to be looking at are precipitation, humidity, temperature, and wind speed. And so what this study actually is, it was a 2-year study conducted along the northern Gulf of Mexico. And we looked at 13 sites, or tidal wetlands, and then within those sites we looked at 3 plots. And we gathered above-ground biomass in 2021 and 2022. And so with that above-ground biomass, we then sorted it into different species, and then we sorted those species by whether they have a C3 or C4 photosynthetic pathway. And so to gather our meteorological data, we utilized 4 proximal weather stations to these sites. And then with that raw meteorological data that we gathered over the 2 years, we then summarized them into annual parameters, such as total precipitation or days of extreme heat or days of extreme rainfall. And so with that, we found that 2021 tended to be a cooler and wetter year than 2022, which was warmer and drier. And so when you're just looking at the average total plant biomass by year, you don't see a clear relationship from 2021 to 2022. It's highly variable and it doesn't tend to increase or decrease with just a raw data analysis. However, when you look at analysis of similarity and you increase your resolution by looking at different species, you do start to see an effect by year. And so this plot that I've included is a non-metric multidimensional scaling plot. And what it does is it takes the similarity matrix of these field plots and it graphs them in 2D space. And so the plots that are closer together have more similarities to each other than the ones that are further apart. And then the lines with the labels, those are our environmental meteorological parameters. And so they are driving the differences in similarities and dissimilarities between the years. And so what we see is that the years tend to separate right off the bat. And when you run a similarity percentage analysis, you find that one species in particular, the needle rush, contributed to 73% of all the similarities between plots in 2021. But when you get to 2022, that contribution drops to about 59%. And you start to see more contribution to similarities from salt marsh grasses in 2022. And when you're looking at C3 and C4 plant biomass, you see a very similar relationship. The plot shows the same separation by years, and it also shows the same environmental meteorological parameters driving these differences. And this also has the same significant effect by year and by site. And so with the similarity percentage analysis, again, you see that contributions from C3 biomass contribute 76% to all the similarities between plots in 2021, but that this drops to 63% in 2022, and you see more contributions from C4 biomass in 2022, which is the warmer and drier year than 2021. And so largely what this can be attributed to, the C3 biomass plants are largely made up of that needle rush species, and the C4 biomass plants are largely made up of those salt marsh grasses. And there's one salt marsh grass in particular, Disticula spicata, which had a very significant increase in biomass from 2021 to 2022. And so what are some of the implications of this? Well, we can see that the hotter, drier weather is more favorable to C4 plants than C3 plants because of their increase in contributions to similarities. And what it can mean for marsh resilience is that annual climatic variability does seem to indicate that there's a stress response occurring in plants. And so when there is a stress response that's happening on an annual basis, a longer-term stressor such as sea level rise could have worse implications for tidal wetlands, and that they could be lost at greater rates over time. Thank you. Are there any questions?
Is it possible that C4 plants will replace C3 plants?
So that is a good question, but with C3 plants, our sites are still dominated by C3 plants like that needle rush. And so it's pretty unlikely that C4 plants would completely replace C3 plants, even if they do have that biological adaptation that makes them better adapted to warmer climates. It would be interesting to see over a longer term study of annual climatic variation how those C3 plants, if they can bounce back from that kind of variation and what they do over the years.
What are some of the next steps in completing this research?
So with what I presented today, I mainly focused on above ground biomass, but we also collected below ground biomass samples and have run some statistical analyses on those as well. And so we want to get some higher resolution in looking at types of below ground biomass that we've collected and separating by different root types and kind of seeing where that goes as well.
Why is there such a difference in the effect by year from the ANOVA to the analysis of similarity?
So that's a great question. The ANOVA is a raw data analysis, and so it's really just looking at the total above ground biomass by year, and it's also a metric analysis. But the ANOSIM, it utilizes pre-treated data that's treated to a similarity matrix and also a Bray-Curtis distribution, which is commonly used for field biomass data. And so it looks at similarities rather than just those raw metric analyses.
You talk about taking above ground biomass samples. What exactly does that sampling process look like? How do you make sure that you get a representative sample of the area that you're trying to collect data on?
That is also a great question. So for each of our sites, we have field plots set up, and I believe that the plots that we sample from are randomly selected within those plots. And so it's not necessarily totally representative of the site, but it's a random selection, so it's the best we can do for that kind of sampling. And also what we do, the above ground biomass, it's just like harvesting the above ground biomass and cutting it and then taking it to the lab to sort and then weigh.